Hello, everyone, and welcome to Cascades Analytics Live. I'm Alexander Stepanovich, founder of Cascades Analytics, a consultancy and media platform focused on the science and future of psychedelics and neuroplastogens. Here we interview the scientists, founders, and investors shaping the next wave of psychedelic-inspired therapeutics. Today, I'm thrilled to welcome Alan Nafiev of Receptor.ai. Alan and his team at Receptor.ai built computational models to better understand how drugs interact with different receptors in the brain. Thanks to recent progress in AI, machine learning has been moving incredibly fast, and we are reaching a new stage in our ability to predict how molecules activate receptors in the brain. Instead of relying mostly on trial and error experiments, which are slow and expensive, Receptor.ai focuses on predicting how a molecule will bind to a specific receptor and what kind of biological response it might trigger before it even reaches the lab. And this is especially interesting in the neuropharmacology of psychedelics because of the 5-HT2A receptor. Small differences in how a compound interacts with this receptor, how strongly it activates it, and how it shapes signaling are closely linked to whether or not it produces psychedelic effects. Being able to model those differences earlier in the process could make a big difference in how neuroplastogens and other brain-active compounds are designed. Hi, Alan, and thank you for being with us today.
Hi, Alexander. Very nice to see you again, and also thanks for invitation. Yeah, very happy to provide you an overview about the current drug discovery landscape and how it's connected to psychedelic industry and where we are from the point of view the best solution that can drive that industry.
All right, great. Well, let's dive in. So, first, from a bird's eye view of the industry, what do you think has improved the most in receptor modeling over the last few years?
Yes, good question. So I think the biggest improvement over the last few years is not a single AI algorithm or model. It's a combination of better data and better ways to explore hypotheses systematically. And we now have much better receptor structures, often in multiple conformational states. We also have better tools to model receptor flexibility and ligand-induced changes. And this allows us to run many more virtual experiments before going to the lab. And as a result, we can explore chemical space more efficiently, compare alternative hypotheses faster, and reduce the number of weak experimental iterations. And importantly, what has improved most is not prediction accuracy alone, but our ability to ask better, more focused questions and test them in a structured way. And I think that shift from single predictions to systematic exploration is what has really changed the field.
Great. So what do you think current receptor models still mislead us?
I think the current receptor models tend to mislead us when we confuse precision with understanding. And we can now predict binding modes and relative affinities quite well in many cases. But in my opinion, that prediction can create false confidence. Models are most misleading when efficacy depends on factors that are not encoded in the structure itself. For example, signaling bias of the receptor, cell type, specificity, timing of activation, and other downstream network effects.
Mhm.
And in these situations, a very clean structure model can still lead to incorrect conclusions about therapeutic outcome. And that's why we're careful not to treat models as oracles. They're powerful tools for narrowing hypotheses, but they don't replace biological validation. The biggest risk today is not that models are bad, but that we ask them questions they are not designed to answer.
Mm. So now a little bit about what you model. What receptors are you mainly modeling today? Still mostly GPCRs, or has that expanded?
So yeah, we are not limited by GPCR family, but we definitely started with GPCRs, and they are still central to our work today, especially in CNS diseases areas. And not because GPCRs are the hardest receptors to model structurally, but because they sit at a very interesting intersection between chemistry, biology, and behavior. And what has really expanded over time is not only the set of receptor families we work on, but the way we think about receptors in general. So we increasingly treat receptors not as isolated molecular type targets, but as controlled entry points into much more complex downstream signaling and network level effects. And this way of thinking applies to GPCRs, and it also applies to ion channels and other receptor families in the CNS. And what matters most for us is not the receptor itself, but whether a short, well-controlled receptor perturbation can lead to long-term biological consequences. And this is especially true in CNS, where transient receptor activation can trigger changes in plasticity, for example, circuit dynamics, and eventually behavior. So when we say we model receptors, uh, what we really mean is that we model the first step in a much longer causal chain. And that perspective strongly influence how we use modeling and how we think about its limitations.
And which modeling approaches are most useful to you right now? And what are the pros and the cons? What are their strengths? And where do they fall short?
Yeah. So, from my perspective, at a high level, the most useful modeling approaches today are the ones that help us understand what parts of the problem we can control and which parts remain fundamentally uncertain. And structure-based modeling is very strong when it comes to receptor engagement. It helps us understand how ligand binds, how selectively they are, and how stabilize certain receptor states. And that part of the problem is relatively mature. And we are now quite good at exploring chemical space around the receptor, ranking compounds with a series, and avoiding off-target liabilities. But when modeling starts to fall short
Mm.
is when efficacy depends less on binding itself and more on how the receptor signals. And in CNS, especially, efficacy often depends on signaling bias, cell type context, and downstream network effects. And no docking score, no single structure, and no static model can tell us which downstream signals are actually causal for a therapeutic effect. And that's why we don't think of modeling as something that solves biology. Instead, we use it as a way to narrow the hypothesis space. And it helps us to avoid, for example, weak chemistry, reduce the number of low quality experimental iterations, and focus experimental work on fewer, better motivated ideas and hypotheses. And in other words, Modeling helps us move faster and more rationally, but it does not replace biology or experimental validation.
So mostly for binding sites.
Yes.
Uh,
You're right.
allostery is already unknown territory. It becomes like maybe the next chapter, and then
So I would
signaling.
say allostery controls a different pathway within the same activation cascade. And sometimes you don't know if that pathway actually correlates with a therapeutic effect you want to achieve. That's why it requires a lot of experimental validation, and this matching between the structure, binding site, different activation cascade, and pathways plus biological response. I would say this is the gaps that we need to fill now. There is solutions that actually can do it. Let's say current LLM systems, different knowledge graph systems that actually combine all of the biological knowledge, all of the entities like drugs, diseases, pathways, genes, omics data, everything interconnected, and you can find transitive and also specific hidden nested correlations and see how you can connect the structure with pathway and the outcome. But again, this is the knowledge driven predictions that can give you hypothesis, but any hypothesis, even if it is predicted by the best LLM in the world, should be validated experimentally. And if the hypothesis is right, then we can build the next step and correct the strategy.
So are there some receptors easier to predict than others? You said GPCRs are probably the hardest.
I would not say that GPCRs are the hardest receptors, but yeah, they're one of the, let's say, promising receptor family from the therapeutic point of view. And of
Right.
course, there is a lot of complexity hidden within that receptors. But anyway, we understand the behavior of these targets. We know the tools that should be applied to work with these proteins. But anyway, what I want to tell you that the structure of the GPCR, but by itself, it's not the main issue. The issue is how that structure and activation of this target translates into the specific therapeutic outcome. And in that case, some receptors are clearly easier to predict than others, and in most cases, this has less to do with the receptor class itself, as I mentioned, and the more to do with how directly receptor engagement maps to therapeutic effect. And receptor tend to be easier when binding and activation are closely linked to efficacy. In those cases, if you understand how a ligand binds, how selective it is, and how strongly it activates the receptors, you already understand a large part of the problem. And these are targets where structure-based modeling and classical pharmacology work relatively well together. And receptors become much harder when efficacy depends on additional layers. For example, when the outcome depends on which signaling pathways are activated, in which cell types, or over what time scale. In CNS especially, small differences in receptor engagement can lead to very different downstream effects. So even if two ligands bind the same receptor, they may produce very different outcomes. That's where predictability drops, not because the structure is poor, but because the biology is more complex.
And is there still some receptors that don't make sense to you, or you can't really work with them, even if there is good data?
Yeah, absolutely. This is important point. There are receptors where we have excellent structural data, for example, high resolution models, and many known ligands, and yet therapeutic outcomes remain difficult to explain or predict. In these cases, the problem is not lack of data. The problem is that structure alone does not capture what ultimately drives efficacy. And this usually happens when therapeutic effect depends on signaling bias, cell type specificity, or downstream network dynamics. And in other words, we can predict binding very well, but binding is not limiting factor. The real uncertainty lies in understanding which downstream consequences are causal for disease modification and which are secondary or compensatory. For example, this is especially common in CNS, where transient receptor activation can trigger long-term change in plasticity and circuit behavior. So this receptor don't not make sense because our models are bad, but because their causal chain from receptor activation to therapeutic outcome is still incomplete, and there is some gap. And this gap between molecular precision and biological meaning is where many of the hardest problems are today.
And so, which disease areas are you most focused on right now?
I would say we are disease agnostic because we approach the problem from the structure based point of view. But from the statistics, what I would say, one of the strongest focus right now is the CNS, and the reason is simple. CNS is where receptor pharmacology connected with the hardest translation problems. And small changes at the receptor level can lead to large differences in outcomes, such as efficacy, side effects, and behavior. And we also have some momentum there. We have multiple publicly announced and non-public collaborations with big pharma companies, clinical stage pharma in CNS. And we have several internal small molecule programs across different target families, including GPCRs and ion channels. And beyond CNS, we engage where receptor signaling is meaningful level, and where structure-based approaches can reduce chemistry risk. But if I summarize our strategy, we focus on areas where better receptor level understanding meaningfully improve decisions upstream before we spend time and money in the lab.
Mm-hmm. And so, where does receptor dysregulation drive disease? Where does receptor modeling help the most in drug discovery?
Yeah, so it's a good question. From my point of view, receptor modeling helps the most when receptor signaling is a clear driver, or at least a controllable driver of disease. In those cases, the link between receptor engagement and biological effect is relatively direct. That's where structure-based approaches are strongest. They help us design for selectivity. They help us avoid off-target liabilities, and they help us prioritize molecules more rationally. And CNS is often a good example because many conditions are treated by modulating receptor signaling. And the difference between helpful and harmful can come from subtle changes in receptor profile, timing, or activation mode. And when modeling helps less, is where receptor signaling is only a small part of a broader pathology. In those situations, chemistry may be the easier part. The harder part is biology and translation. So the right expectation is modeling reduces chemistry risk, but it does not eliminate biological uncertainty.
Mhm. So, to what extent, like diseases like cancer or heart disease, inflammation or kidney disease, driven by receptor signaling versus other factors like, uh, like I don't know, tissue damage or metabolism?
Yeah. So it depends a lot by indication, and it's important to be realistic about that.
Hmm.
In cancer, receptor signaling can be a strong driver
Hmm.
in specific context, but
Hmm.
it's often embedded in genetics, cell state, for example, and microenvironment. So receptor modulation can help, but it's rarely the whole story. Uh,
Hmm.
um, in inflammation, for example, receptor signaling can be very central, but outcomes depend on the immune network, feedback loops, and tissue context. So, again, signaling is important, but it's a part of larger system. And in heart and kidney disease, regulator signaling absolutely matters. And but disease progression is often dominated by tissue remodeling, damage, and systemic physiology. So modeling can still be valuable, especially for selectivity and safety, but it won't solve the indication by itself. So the way we think about it is this: receptor modeling is most impactful when it helps us control one meaningful lever in a complex disease, and reduces the cost of exploring different hypotheses and options. But we always align expectations. Structure and AI reduce chemistry risk. Biology and clinical translation remains currently the bottleneck.
Hmm. Hmm. Well, thank you. That's fascinating. Let's move to psychedelics now. So, have you looked at the 5-HT2A receptor at all? And yeah, I'm curious because that's the 5-HT2A is one big question in the psychedelic field right now. So,
Yeah, we have couple of collaborations
mhm,
with different companies from CNS psychedelics industry, and yeah, we know that receptor very well, and such as receptor like 5-HT2A, very well covered by existing and structural pharmacological data,
mhm,
and modeling at that level is quite mature. Modeling can realistically answer questions related to receptor engagement. For example, how different ligands bind, how selective they are relative to closely related receptors, for example, and how they stabilize different receptor conformations. That part of the problem is relatively well understood today.
mhm.
So, where modeling becomes more limited is when we move beyond receptor engagement and
Right.
start asking questions about therapeutic outcome. In that case of psychedelics, the core challenge is not whether a compound activates 5-HT2A, but which aspects of that activation lead to durable therapeutic benefit versus the subjective psychedelic experience. And structure based models can help us explore different modes of engagement, uh,
Mhm,
such as partial agonism or potential signaling bias. but they cannot by themselves tell downstream effects are causal for therapy, for example. So we see modeling as a tool to design and prioritize hypotheses at the receptor level, as a way to directly predict clinical or behavioral effects.
Right. Any interest in trying to discover new binding sites, allosteric binding sites, maybe?
So yeah, there is a lot of work around finding and discovering the allosteric binding sites, and we're also engaged in that work. So this is not, let's say, trivial. That's why we apply different strategies, translocation of the allosteric sites from other known GPCR receptors and from the very similar families, and sometimes such a strategy works. Sometimes we apply very intelligent and smart techniques like AI combined with physics-based approaches that evaluate the receptor conformational changes to see if the allosteric pocket is exist and if it's opening and closing during the dynamics. In that way, sometimes we can achieve the selectivity when we're targeting allosteric pockets. In such way, we reduce off-target effects and minimizing the interactions with other isoforms of the same family. And in addition to that, allosteric modulation can open for us kind of activation of specific pathway selectively. But again, the problem is how to match this activation pathway to a favorable therapeutic effect without psychedelic experience. This is the challenge now. So
Right.
usually it's trial and errors, and so it's where we are currently.
And are you exploring the polypharmacology of psychedelics? I'm thinking about LSD, for example, that is known to hit the 5-HT2A, but also a myriad of other receptors. So, are you using these compounds to understand the interaction with multiple receptors at once, or do you focus mostly on one receptor at the time?
Yeah, this is a very good question. And yes, polypharmacology is a central consideration for us, especially in CNS. And most psychedelics are not single receptor drugs. They interact with multiple serotonin receptors and sometimes additional targets as well. From our perspective, this multi receptor profile is not a complication to ignore, but it's rather a property that needs to be understood and shaped deliberately. And modeling help us map and compare receptor interaction profiles, identify potential of target risks, and explore how different combinations of receptor engagement may contribute to efficacy or side effects. And at the same time, we're careful not to overinterpret these profiles. Knowing that a compound binds multiple receptor does not automatically explain its therapeutic effects. So the real value of polypharmacology analysis is in narrowing the design space and avoiding obvious liabilities rather than claiming full mechanistic understanding. In that case, polypharmacology modeling is about risk reduction and the rational exploration, and not about predicting complex clinical outcomes. We even now have the project with the clinical stage company in the CNS area, and this is publicly announced collaboration where we actually trying to predict and identify the target protein engaged in specific CNS disease. So they have the molecule, they have the phenotypic effect, but they don't know where the compound binds. And we started doing our research, and we also identified that this compound binds to multiple receptors. That's why sometimes, as I mentioned before, within CNS indication, usually compounds actually activating multiple receptors, because there is a lot of isoforms that are similar and share very similar structural characteristics. That's why, yeah, we know about that issue, and we have technologies that actually can help narrow down the mechanisms of activation and demonstrate where the compound binds from the structural perspective. But then the question how to map it to the biological response, this is the most, let's say, active complex difficulties within our area and within psychedelic industry.
Mhm. And then there's one, burning question right now is the question of receptor occupancy and activation time, especially when it comes to psychedelic effects. Is that something you've worked on?
So yeah, in this area where psychedelics really challenge traditional pharmacology assumptions, in many therapeutic areas, receptor occupancy correlates reasonably well with efficacy. In psychedelics, that relationship is much less clear. So short periods of receptor activation can lead to effects that last weeks or months. At the same time, high occupancy or prolonged activation does not necessarily translate into better outcomes. And this suggests that timing, signal dynamics, and downstream plasticity are at least as important as binding strengths. So, from that perspective, occupancy alone is not a sufficient metric to explain psychedelic effects. The key questions are more about how long the receptor is activated, in what context, and what downstream programs are triggered as a result. That's also why designing scalable drug-like psychedelics is so challenging. And we're not simply optimizing for stronger binding, but trying to understand what the minimal and controllable pharmacological intervention is that to produce durable benefit.
Mhm, mhm, mhm. Now, if we think a little bit, you mentioned that already, but if we, I wanna know a little bit more about that gap between receptor understanding and or receptor prediction of its activity to integrate it to behavior. So, how do you think about bridging the gap between receptor level activity and real changes in mood or behavior?
Yeah. So from our perspective, we think about this gap actively
Mhm.
as a multi-step causal chain. And at one end, you have receptor engagement and signaling. On the other hand, you have changes in mood, perception, or behavior.
Mhm.
And between those 2 points, there are many layers and levels. For example, downstream pathways, cell type specific responses, circuit level effects, and plasticity. And structure based modeling is strongest at the very first step of the chain, bridging the behavior requires us integrating receptor-level perturbations with some biological readouts and network-level analysis. So from our perspective, the goal is not to directly predict behavior from structure. The goal is to identify which receptor-level signals are necessary and sufficient to trigger the downstream changes that matter therapeutically. That's why we think in terms of causality and hypothesis testing rather than end-to-end prediction.
So now, if we look at the the drug as hitting multiple receptors at once, so many CNS drugs hit multiple receptors. So when do you think models will reliably predict the effects of drugs with complex multi-receptor profiles?
Yeah, so I think models are already useful today for certain aspects of multi-receptor drugs. They can help us design and compare multi-receptor engagement profiles, for example, avoid obvious off-target risks, and reason about relative selectivity. Where models are not yet reliable, I think is in predicting full clinical or behavioral outcomes from complex multi-receptor interactions. That limitation is not just technical, it's biological. The effects of multi-receptor drugs depend on context, timing, and downstream network responses that are not fully understood. So I don't think that the question is when models will predict everything reliably. I think the more realistic trajectory is that models will increasingly help us design better hypotheses and reduce uncertainty, while biology and clinical data remain essential for understanding outcomes. So, in other words, models will guide decisions, and but they won't replace experimentation.
Well, thank you very much, Alan, for this contribution that was fascinating. And thank you all for tuning in. If you have any question, feel free to DM me. And I'll see you soon on the next live show of Cascades Analytics. Thank you. Have a great weekend.